Hello, and welcome to this GONE Network webinar. Today, I'm very pleased to be able to share with you a comprehensive overview of the progress of the development of Moxidectin, a promising additional tool to help accelerate the elimination of onchocerciasis. Over the course of this presentation, I'd like to share with you details of the development of Moxidectin, its current status, and the anticipated near-term next steps to achieving recommendations for its use. But firstly, I'll introduce you to the Medicines Development for Global Health organisation and the medicines and diseases we're working on. I'll then move on to explaining the key characteristics of Moxidectin and present data from the phase 3 study that supported its approval for treatment of onchocerciasis. I'll then cover the work that's been ongoing to provide the necessary data to support policy recommendations and the next steps that we intend to take to facilitate access to Moxidectin for use in onchocerciasis treatment programs. Medicines Development for Global Health is a product development partnership with unique characteristics. Like other PDPs, Medicines Development collaborates in the development of new medicines for global health needs. Medicines Development's a non-profit organisation, and we rely on grants, philanthropy and impact investment to do our work. However, unlike other PDPs, Medicines Development holds the licence and therefore the regulatory responsibility for the medicines we develop. You'll see that the Medicines Development brand appears on the tablet bottle labels. We're just a small team at Medicines Development, only 30 people, and so it keeps us very busy while we're running several development programs for 2 molecules. Medicines Development is currently actively targeting 4 categories of parasite-causing diseases: the filarial diseases onchocerciasis and lymphatic filariasis, ectoparasites, scabies and head lice, the helminthic worm infections strongyloides and soil-transmitted helminths, and mycobacterial infections, including leprosy type 2 reactions and tuberculosis. Collectively, these diseases affect more than 1 in 5 people on the planet, usually in the most impoverished nations. Our main priorities for the Medicines Development team is in the development of these medicines for 4 of the 8 diseases, including onchocerciasis, LF, scabies, and leprosy type 2 reaction. We're working in partnership with other clinical partners in the development for those other diseases that I mentioned. The development of Moxidectin for onchocerciasis and other human diseases has been touched by many people and organisations, beginning with the original developer, Wyeth, who worked in partnership with the WHO Special Program for Research and Training, the WHO TDR. Medicines Development became involved in the development of Moxidectin in 2014, and we worked to successfully register the product with the United States Food and Drug Administration in 2018. This slide summarises the data that were provided to support the initial approval of Moxidectin for onchocerciasis. Much of the research was conducted by the previous sponsor, Wyeth, added to by Medicines Development, and brought together in a comprehensive regulatory submission. In addition to all the data required to demonstrate the quality of the Moxidectin tablets, data from many non-clinical mechanistic and safety studies, as well as 6 phase 1 studies and the pivotal phase 2 and the phase 3 clinical trials in onchocerciasis were included. As you can see, the phase 2 and 3 studies were conducted in Ghana, and the phase 3 study was also conducted in the DRC and Liberia. The approval was granted for treatment of onchocerciasis in people aged 12 years and older. Moxidectin is a second-generation macrocyclic lactone. It has a similar structure and mode of action to ivermectin, but with some key characteristics that differ from ivermectin. One of these key characteristics is its very long pharmacokinetic half-life, meaning that moxidectin is retained in the body for much longer compared with ivermectin. Moxidectin is stored in body fat and gradually released back into the circulation. The continued presence of moxidectin in the body enables the sustained suppression of the Onchocerca microfilariae. Let's explore the evidence for moxidectin's sustained effect on the Onchocerca volvulus microfilariae. This is the design of the pivotal phase 3 clinical trial. Adults and adolescents 12 years of age and older, and with at least 10 microfilariae per milligram of skin before treatment, were enrolled at 4 sites in Liberia, Ghana, and the DRC. They were treated with either a single dose of moxidectin 8 milligrams or ivermectin 150 micrograms per kilogram. You'll note that there are twice as many people who received moxidectin as ivermectin. They were followed for 6 days after treatment in an inpatient period and had their skin microfilariae measured at month 1, month 6, month 12, and at month 18. Safety was assessed by adverse events, physical examination, and assessment of Mazzotti reactions, as well as blood measurements. This slide shows the series of scatter plots that depict the impact of treatment on microfilarial density at each of the 4 time points after treatment. The x-axis of each plot represents the density of microfilariae in the skin of each individual before treatment. The y-axis represents the density of microfilariae after treatment at each of the 4 time points, month 1, month 6, month 12, and month 18. You can see the results for ivermectin in the top 4 panels, labelled A to D, and for moxidectin in the bottom 4 panels, labelled E to H. Each point represents an individual enrolled from one of the 4 centres, as represented by the symbols. Recruitment was from Nord Kivu and Nord Ituri in DRC, Lofa County in Liberia, and Nakwanta District in Ghana, areas that had not previously received prior CDTI. As you can see, more individuals in the moxidectin treatment group achieved very low levels of skin microfilariae at month 1, and by month 6, the proportion positive for skin microfilariae had decreased even further. This slide highlights the proportion of participants with detectable levels of skin microfilariae at each time point measured in the study for either treatment. These data indicate that moxidectin offers a more complete and longer-lasting reduction in microfilarial density in infected individuals compared with ivermectin. This could have significant implications for the elimination of onchocerciasis, particularly where this has not yet occurred despite ongoing treatment with ivermectin. You'll note that at 12 months after treatment, the number of people with detectable infection in the moxidectin group remained lower compared with 6 months and even 1 month after ivermectin. If we think about the phase 3 study data another way, if we take 100 people in each of these 2 boxes with high levels of skin microfilariae and treat them all, 6 months after treatment with moxidectin, only 9 of the 100 people would have detectable skin microfilariae, and each of those that did have it would have very low levels, compared with the ivermectin group on the right. Having no skin microfilariae available when black flies bite means that there will be far less opportunity for infection to be transmitted from person to person in the community, thus reducing or even preventing the establishment of new infections. Thus, persistence of infection within an endemic community will be reduced. At 12 months after treatment, the greater proportion of people with undetectable levels of skin microfilariae and overall lower burden of skin MF will also contribute to reducing both transmission of disease as well as the burden of onchocerciasis on the people who have the infection. So let's move on to the safety of moxidectin. In healthy volunteers, the profile of adverse events has been shown to be similar to placebo. And in people with onchocerciasis, the most commonly reported adverse events were related to the efficacy of treatment, resulting from the immune responses to the dying microfilariae, and similar to the events seen with ivermectin. Overall, efficacy-associated adverse events were no more severe with Moxidectin than with ivermectin. None of the serious adverse events reported in any of the studies conducted to date have been assessed as drug-related. On this slide, you can see that the time to resolution of efficacy-related events after Moxidectin was also similar to ivermectin. These are the results from the phase 3 study, showing that the majority of events commenced and were resolved within the first week after treatment. Ocular events were studied closely in both the phase 2 and phase 3 studies of Moxidectin compared with ivermectin. You see here the results from the phase 3 trial. As you can see, similar proportions of events occurred with either treatment. All ocular events were graded as mild or moderate in severity. There are a few key things to note about Moxidectin tablets. Moxidectin is administered according to age rather than weight or height. The tablets can be taken with or without food. And as you can see, the tablets are oval. They are also quite small, about the same size as ivermectin tablets. Each tablet contains 2 milligrams of Moxidectin. So a dose of 8 milligrams, as we talked about in the phase 3 study, is 4 tablets. Like ivermectin, the Moxidectin prescribing information includes precautions for the use of Moxidectin where lower is co-endemic, or to screen individuals who may have been exposed. Further data are currently being generated to better understand the safety of Moxidectin in lower infection. In addition, people with hyperreactive oncodermatitis, also known as SAUDA, are advised they may be more likely to experience edema and worsening of oncodermatitis with Moxidectin treatment. There are some special populations that need to be considered when deciding treatment with Moxidectin. Like ivermectin, pregnant women are advised not to take Moxidectin during their pregnancy. For breastfeeding women, the US FDA approval recommends not for breastfeeding at the time of treatment and for 7 days after treatment. Further analyses have been conducted, and the information that we generated has now been published in a paper in PLOS NTD. These new data will provide a basis on which public health decision makers can make recommendations for use of Moxidectin during program use. The data could support a recommendation similar to that of ivermectin. Lastly, no dose adjustment for Moxidectin is necessary for the elderly, nor for people who have reduced kidney or liver function. Since achieving the initial approval for Moxidectin, Medicines Development has been undertaking further studies to complete the package of data and information needed for World Health Organization recommendation. We've been working collaboratively towards completing the package of data needed for the WHO filarial treatment guidelines. These data are almost complete. Data from the initial studies I've talked about, and many recent studies in children and in typical onchocerciasis and LF co-endemic populations, as well as data gathered in other neglected tropical diseases, will provide comprehensive data for efficacy and safety on which to base the guidelines recommendation. Epidemiological and cost modelling has been done and continues to be generated. Lastly, acceptability, feasibility and community preferences will soon be assessed through social research conducted alongside Mass Drug Administration pilot programs with Moxidectin in endemic communities in Ghana and Angola. I'd like to now provide you with an overview of the more recent data and the planned activities in support of the guidelines, and to facilitate implementation in country programs. Since the initial FDA approval, we've initiated studies to generate field-relevant data and to expand the approved label to include children down to the age of 4 years, thus covering all relevant age groups for MDA programs. A pharmacokinetic and safety study in children was completed last year, which led to a protocol amendment to include children 4 years and over in a large ongoing safety study in the DRC and Côte d'Ivoire, where onchocerciasis and at one site, LF, are endemic. More than 13,000 people have been enrolled, including more than 1,000 children, approximately 80% of whom have received treatment with Moxidectin, the other 20% with ivermectin. A study of annual or biannual treatment with Moxidectin or ivermectin is ongoing. Each participant will receive treatment with either 3 doses of annual or 5 doses of biannual treatment with either Moxidectin or ivermectin over 2 years, with 1 year of follow-up. The data from this study will further inform the epidemiological modelling of use cases and elimination outcomes that will inform programmatic decisions. Data from the paediatric and large safety study mean we are now well placed to implement small-scale community distribution projects to assess the acceptability and feasibility of MDA with Moxidectin. This is the design of the paediatric PK and safety study. Healthy children and adolescents were recruited from communities in Nakwanta North District in the Oti region of Ghana, which is onchocerciasis endemic. These communities were chosen where previous studies had been conducted so that community members were familiar with research concepts and procedures. Cohort 1 included adolescents 12 to 17 years old who received the approved dose of 8 milligrams. Cohort 2 included children 8 to 11 years old. They also received an 8 milligram dose in the first group. That dose was selected based on a population pharmacokinetic model developed from the data in healthy and ovovolus infected adults who received single Moxidectin doses ranging from 2 to 36 milligrams. The PK and safety data collected up to 28 days after treatment were reviewed by a DSMB, or a Data Safety Monitoring Board. Evaluation of a 6 milligram dose in 9 additional 8 to 11-year-old children was also undertaken as a protocol mandated requirement based on the PK results in the first group of 8 to 11-year-olds. Upon review of all the data from the first 2 cohorts, the DSMB agreed that enrolment and dosing of 4 to 7-year-old children with Moxidectin 4 milligrams could be appropriately undertaken in parallel with the second group of 8 to 11-year-olds receiving 6 milligrams. This is the disposition of the study. 100 potential participants were pre-screened in their communities, and 66 completed full screening at the study centre. To minimise any safety risks, children in cohorts 2A and 3 were dosed in subgroups. A three participant sentinel group who received treatment first, and then the remaining 6 participants. Data from all 36 participants of different ages receiving their allocated doses contributed data to the modelling that was then used to select an appropriate dose for children across the age range of 4 to under 12 years. These are the adverse events for all 4 cohorts of children, collected from day 0 of dosing to week 24 or 6 months after treatment. The adverse event data show that Moxidectin was well tolerated by all the participants, independent of dose or age group. No serious adverse events were recorded, and none of the adverse events were considered Moxidectin related. The types of AEs seen represent those expected by the study team in children in the area over a period of 24 weeks. In summary, Moxidectin doses of 6 or 8 milligrams were well tolerated in the 8 to 11-year-old children, and a 4 milligram dose was well tolerated in 4 to 7-year-old children. Although different age brackets and weight brackets were assessed, further analysis suggests that dose selection can be simplified to an 8 milligram dose for 8 years and older, and 4 milligrams or 2 times the 2 milligram tablet for 4 to 7-year-olds. As mentioned, more than 1,000 children enrolled and treated in the large safety study reaching completion now in DRC and Côte d'Ivoire were treated according to this 2-tiered dosing regimen. The outcome of submissions to the US FDA and the Ghana FDA will confirm the dose for children. Epidemiological modelling has shown that Moxidectin has the potential to contribute to greater probabilities of elimination and with fewer rounds of treatment. The figure on the left displays the projected number of years to achieve a 90% probability of eliminating transmission, showing that Moxidectin significantly reduces the time needed to reach elimination compared to ivermectin given at the same frequency. 4 treatment regimens have been compared: annual or biannual Moxidectin, shown here in blue and labelled as ACDTM, shown in the solid colour, or BCDTM, shown with hatching, and annual or biannual ivermectin, shown in the red bars and labelled similarly as CDTI and BCDTI. The right figure highlights the relative programmatic delivery costs for the different strategies compared with annual ivermectin, indicating substantial cost savings on program implementation could be made. As an example, in an area with a baseline onchocerciasis prevalence of 50%, shown in the middle bars in the left panel, annual Moxidectin, in the solid blue bar, would reduce the time to elimination by 50% compared with annual ivermectin, leading to a 36% programmatic cost reduction, shown in the middle bars in the right panel. In areas with a 30% baseline prevalence, in the lower bars, the time to elimination would be reduced by 8 years using annual Moxidectin compared with annual ivermectin, resulting in a 33% cost reduction in program costs. A field implementation project named Momentum is well underway in planning in Ghana. The objective of this project is to generate relevant data and experience to support the introduction and use of Moxidectin in onchocerciasis elimination programs. Twice yearly MDAs with Moxidectin are planned for 3 years or 6 rounds in the Twifo-Ati-Morkwa district of Ghana. This region was hoping to confirm stop MDA, but as a result of continuing prevalence has had to restart treatment. This project will also confirm the feasibility of implementing Moxidectin as an alternative to ivermectin and will assess the acceptability of this new treatment by the local population and stakeholders. Training and education materials will also be road tested. The efficacy of Moxidectin in lymphatic filariasis has also been assessed in a clinical study conducted in Côte d'Ivoire. In that trial, Moxidectin plus albendazole, or MOX-A, was shown to be superior to ivermectin plus albendazole, IA, and similar to the triple combination of ivermectin, DEC, and albendazole, or IDA, the current gold standard for treatment of LF in areas not co-endemic for onchocerciasis. The data on safety for Moxidectin combination treatment in this study showed that adverse events were mild, self-limited, and did not differ between IA and MOX-A. The large community safety study I mentioned earlier will provide more information on the safety of the MOX-A combination in adults and children who live in onchocerciasis and LF co-endemic communities. A follow-on study in those shown to be LF infected before treatment will provide further data on the efficacy of Moxidectin and albendazole in that disease. Other studies in LF are planned. Given the co-endemicity of onchocerciasis and LF in many countries in Africa, the data so far suggests there'll be an opportunity to use Moxidectin for the treatment of LF as well as onchocerciasis in co-endemic settings, providing potential synergies for programs and with the potential to address challenges in the elimination of both diseases. So what are the next steps for medicines development to facilitate access to Moxidectin in endemic countries? You may be aware that the World Health Organization recently triggered the process for the development of treatment guidelines for treatment of onchocerciasis and lymphatic filariasis with Moxidectin. In parallel with that activity, Medicines Development is planning to undertake a number of steps to make Moxidectin accessible and available to endemic countries. The first step will be to submit to the WHO Essential Medicines listing, the next round of review. With the issuing of an expression of interest, Medicines Development will also submit to the WHO for pre-qualification. We're also investigating how to support appropriate authorisations for use in endemic countries. We're also keen to work with countries and partners and stakeholders along with the Mectizan donation program, to understand where Moxidectin could add value and how we should coordinate treatment. We're looking to scale up manufacturing according to the need, with support from donors. And lastly, we want to develop with partners a sustainable funding model to support access to Moxidectin for the longer term. This slide shows key product use specifications for Moxidectin 2 mg tablets in 500 count bottles. The current specifications are presented in column 2 and the target future changes in column 3. Our intention is to optimise the characteristics on the approved label that will support optimal product use in country and its use in MDA treatment programs. As I've already mentioned, we're intending to seek regulatory approval for the treatment of onchocerciasis from 4 years of age. We're also planning to generate the necessary data to increase both the in-use or open bottle use period and the shelf life as much as possible. We'll also be working to generate the data to support its use in the hottest and most humid climatic conditions, known as Zone 4B conditions. We anticipate that the current and future use will also be informed by WHO review and policy recommendation. And finally, I'd like to thank and acknowledge, on behalf of the whole medicines development team, all of our partners and the countries that have been involved in generating the data necessary to define how and where Moxidectin might be used, and to what extent it could contribute to countries' onchocerciasis and LF elimination efforts. We look forward to working with you further, and thank you for all your efforts and contributions. And thank you for your participation today. If you have any questions, Medicines Development for Global Health, in partnership with GON, would like to invite your participation in a Q&A session to be held on the 20th of August. An invitation email for registration to this GON event will be circulated soon, if not already. To facilitate responses to your questions, we invite you to submit them via an email to barbara.roth@medicinesdevelopment.com. In addition, you may find it helpful to look up the Medicines Development website at the link noted on the screen. Until then, I wish you the very best and look forward to seeing you soon. Bye for now.